for this presentation. And today I'm going to speak about a topic called biomarker-based drug combinations. And in that case, we are exploring epigenetic vulnerabilities, one of the anchor space. And so my previous speaker, Dr. Dhruv, has already actually made my life easier because he has already kind of explained the importance of combinations. So there is actually a nice paper called It's Combination Stupid. So it's kind of, we need combination. Without combinations, if you just go for single agent drugs, it's not gonna, we are not gonna actually control the tumor. So basically, we need combination and we need rational combinations. So there are a lot of single agent drugs which are effective on their own way, but you need to find rational combinations out of these thousands of drugs. So you need to find a way how you can rationally approach and find these combinations. So here I will try to give an example with some indications how we can make use of different strategy to find new combinations and take it to the clinic. So basically there are a few things you need to know. So it's about combinations, different drug combinations. It could be small molecule, large molecules, antibodies, anything you can work on. And also to actually get nice combinations or kind of rational combinations, you need to have certain number of tools to actually do this kind of experiment. And also you need to have a good model systems. And then you have to actually start with some kind of indications. And our indications of choice is actually we started we are kind of a lab with more work on the lung cancer field, and mesothelioma is one of the kind of lung cancer which we work on. So many of you might be wondering what is this disease called mesothelioma. Okay, so this is a asbestos-induced cancer of lungs, and it's called mesothelioma. It specifically originated in the mesothelial layers of the lungs. So if you look at here, say this is just normal lung, and in the normal lungs there are actually layers of cells or layers of tissues called mesothelial layers. It's called mesothelial tissues. If the tumors specifically originates in these layers, these tumors are called mesothelioma. And this is actually a very nasty tumor. If it starts here, actually grows so much, actually it fills up your whole thoracic cavity, and it compresses the lungs, and actually people with mesothelioma, they will have issues with breathing and chest problem, a lot of tumors. Like in terms of tumor, you can find many kilograms of tumor inside your body. So it's a kind of and this is actually a very, this is very relevant in terms of India because this is actually specifically happens because of the asbestos exposure. Anybody who are kind of long-term exposed to asbestos, they develop this kind of tumors, but I think the frequency is still quite low. And if you look at in India, a lot of kind of places, we are still using asbestos, and it's heavily used. And if you look at a lot of Western countries, even though they have used in the past, but now they have completely banned using asbestos. But if you go to our rural India, almost everybody is making a roof of this asbestos because it's cheap and durable. So this is a very alarming situation. So what is this tumor? So this mesothelioma is very aggressive. It grows very locally and aggressively, and there are many different histological subtypes that you probably come across. And it is a very poor prognosis. Obviously, many diseases poor prognosis. So far, you can say chemotherapy and immunotherapy has been approved. But if you look at their kind of success rate, is very minimal. I'll come back to this slide, the next slide. So there is an urgent need of better treatment modality. Just to give an idea what is available in terms of chemotherapy and immunotherapy. If you look at, this is kind of overall survival of the patient, and this is kind of how many months after diagnosis somebody can actually live. If you go for it, say for example chemotherapy, which is actually this red line over here, most of the patients, say for example the minimal, so they survive certain amount of time. So for example around 8 to kind of 14 months is the very average survival if you go undergo chemotherapy. But if you, on top of that, like if you choose for say immunotherapy, which is quite expensive one, you might survive another 4 months more, like 18 months, you can say. and this is still kind of very dismal. So we need better treatment or kind of combination of treatment along with either chemotherapy or this immunotherapy, so that we can actually kind of at least extend the survival of these patients. So to undergo this one, so we need a lot of tools, as I mentioned. So you need to have right tool to do this kind of experiments. So what we did in our lab, we actually generated a mouse model and also generated a lot of cell lines out of this, so that we can do many combinatorial drug screening, CRISPR screening in this. So even to understand any disease, you need to first understand the genetics of the disease. So to give an idea of what is the genetics looks like in mesothelioma. So in mesothelioma, there are a lot of genes mutated. So for example, we have some of the gene mutations. One gene called BAP1 is called BAP1. BAP1 stands for BRCA1 associated protein 1. So it's kind of linked to BRCA1. There is another, and which is actually a part of a polycomb repressive complex, or kind of a PR-DOP complex, polycomb repressive D-ubiquitinase complex. Then another gene is called NF2. NF2 stands for neurofibromatosis type 2 genes. And this is an important regulator of hyposignaling pathway and Merlin signaling pathway. The third most common gene actually over here is called CDKN2. Over here you can see this, you see mostly the copy number changes. And CDKN2 is a gene mostly, is a regulator of cell cycle. Yeah? And if you look at these mutations, most of these mutations you see are mostly in the tumor suppressor. You hardly see any oncogenic events, specifically in mesothelioma. And so it's very difficult to actually target tumor suppressor. If there is oncogene, there is many people actually target oncogenes, but targeting tumor suppressor is very difficult. So you need to find a different way of targeting these tumor suppressors. So what we did, we took advantage of these genes, mutations or alterations, and make a mouse model out of it. So specifically we incorporated these highly frequent genes like BAP1, NF2, and this CDKN2 in a mouse model. We incorporated, what's the incorporated means? We try to delete these genes inside the mouse, specifically in the lungs area, mesothelial area. And how do we do this one? First, what we do is we use a technology called Krelux technology. I think many of those who work on mouse or mouse model or genetically engineered mouse model, they know this one. It's called Krelux technology. You put a Krelux, kind of a Lux site on kind of around these genes, BAP1, NF2, or CDK2 genes, for example, over here. And then by recombinase, you can kind of do a recombination, and that's how you can actually delete this part of the gene, and you can have a defective gene product, or maybe there is no protein at all of these genes. And what we did, we do kind of introduce this allele inside the mice, and then we delete this by external virus. The virus containing recombinase goes into the lung, and then actually delete these genes, and eventually these genes are deleted, and the mouse develop tumors, and they are specifically developed mesothelioma. If you look at this, it's a kind of the lungs area over here. This is a small tumor, and this is kind of the thoracic cavity, and you can see in the thoracic cage, this white area is kind of tumor patch. And so we did actually deletion of this gene in different combinations. We did try to delete all the three, or in combination with BAP1 or kind of NF2, or many different combinations. So just to give an example, here is a combination. When we delete NF2 and CDKN2, these mice develop tumor, although it takes some time, like it takes almost 6 months, median say incidence time is like 6 months. But when you on top of that delete BAP1 gene, you do a very rapid kind of see a rapid tumor development. So most of the mice develop tumor between 2 to 3 months, and they die actually after the time span. So this model is kind of an ideal model system in the sense you have a very rapid tumor development, and it mimics a lot of features of human mesothelioma. Just to give an example, these tumors are also highly invasive like the human mesothelioma, and fast tumor development. It recapitulates signaling pathways, immune profiles, therapy response, and also closely mimic human mesothelioma. So how do you believe me that they are kind of mimic all these features? Just to give an example, say for example, the human mesothelioma, they develop tumors from kind of epithelial or sarcomata and mesothelioma. these mouse also develop this kind of mesothelioma. And the important feature of this mesothelioma is that, say, it's epithelial kind. You see kind of, say, vacuolated cells. You see also vacuolated cells over here. In sarcomata, the cells are arranged in bundles and spindle-shaped cells. You do also see over here in this mouse too. So they closely mimic in terms of histology and also histotypes. And we extensively characterize these tumors. I have not actually gone through many of these markers. So one of the markers is actually WT1. The brown cells over here are the staining of these tumor cells with this particular marker. And this is kind of the mesothelioma. So how fast they grow? So these tumors actually grow as fast as you can think. So for example, if you do a kind of a xenograft experiment, within 3 to 4 weeks, we generally get tumors. And this, even though it's a kind of an immunocompetent and model, autochthonous model, it do develop so fast. Even within 4 weeks after deleting these genes, you can already start seeing incidence of tumors. So this is a normal mesothelial lining around the lungs. But if you look at within 4 weeks, see how much of kind of hyperplasia kind of tumors incidence you can already see. How thick are these layers? Within 4 weeks of deleting these genes. This is another layer of mesothelial layer on the diaphragm area. You can see that how thick is this already tumor cells are. And these tumor areas already show all the pathways like for example, MET kinase pathway, PI3 kinase pathway, everything all together. So this is a very ideal model. You can actually use this model for therapy response, and we have actually used this one. I'll come back to that in the later. But other things you can see, it do also mimic quite substantially in the inflammatory or tumor microenvironment. So if you look at these are the different humans, for example, is a macrophage staining, the T cells, T-rex cell staining, or just normal T cell staining. You see kind of very scattered number of these kind of cells, and you do see also in the mouse these tumor cells are present. And we have kind of extensively characterized this by looking at the RNA-seq data from these, and you can see that there is a kind of a special differences in this kind of macrophages and macrophages over here. So this is not the focus of my talk. My focus is mostly on the combinations. So I'll quickly go to the combinations. But before going to this, whether this model that I'm talking about, is it a suitable model to do therapy? Or can you use this model directly as a therapy purpose? So what we did, so we induce tumor, and then after, say, for example, 6 weeks, we give a dose of drugs here, and the second dose of drugs. And which drug we use? We use the drugs which is actually is a very standard therapy drugs. In mesothelioma, most of the people, they get cisplatin plus pemetrexed as the first line therapy. So that's what we use, and in this particular model, we use these drugs and see whether we can get a response. And we do see kind of a significant survival improvement. So they roughly survive 3 weeks more than untreated mice. And this is actually quite substantial amount of time, looking at the age of the mice. And you do see kind of a substantial reductions in, say, PI60, this is a kind of proliferative cells and the cell death, especially marker. So this tells us this model is very suitable for doing therapy. So, as I mentioned, I will talk something about the biomarker. What kind of biomarker you can use for this kind of therapy response. And specifically, I'm choosing this BARC1 because BARC1 is a part of a complex. This is called PR-DOG complex. PR-DOG means polycomb repressive D-ubiquitinase complex. So what this complex do, it removes ubiquitin from H2A119. If you remove ubiquitin from H2A119, it actually opens up the chromatin, and that's why you can kind of have more gene expression. So for example, over here, so if you remove this ubiquitin mark over here, the chromatin complex will be open, you will have more expression, and then more expression of downstream genes. But the opposite of, like for example, the PRC1 or PRC2 complex, these are the two complexes which helps in putting either ubiquitination mark or S3K27 trimethylation mark, and these are more repressive chromatin mark. This actually close the chromatin more, and then you have more gene repressive chromatin. So the BARC1, what it does, it does opposite things what the PRC complex do. And we have seen that this BARC1 protein has been actually, a gene has been mutated in many different cancer types, and specifically mesothelioma, uveal melanoma, renal cell carcinoma, cholangiocarcinoma. So this will be kind of an interesting subject or interesting kind of indications where we can use BARC1 as a biomarker for patient stratifications or maybe a therapy response. So what does actually in terms of molecular aspect of BARC1 do? In these studies what we did, we took NC, where the NC stands for NF2-NCTK2 deleted cells, and BNC stands for BARC1 on top of the NC. So the difference between these two is kind of BARC1 difference. So it's kind of a isogenic kind of cell line. The difference is BARC1 deletion. If you look at the chromatin, or say for example S3K27 trimethylation binding, if you look at the promoter area, so in case of BNC, there are a lot of binding in the transcription start sites. So that leads to more gene repression. And that's why in BNC cells, we do see a kind of a repressive chromatin loss, in the sense that like almost 70% of the genes are downregulated as compared to kind of normal cells or normal expression. So it means this S3K27 trimethylation gives a repressive down transcriptional program. A lot of genes are downregulated. But in that situation also there is a gene called EGS2, which is actually specifically upregulated, which is a part of the PRC2 complex. And we kind of make use of this EGS2 overexpression. Can we kind of shut down the EGS2 expression and kind of make this cell sensitive to, say for example, killing? And we did some experiment on this one. Say for example, if you take this BARC1 deleted cells over here and knock down an EGS2, you can easily kill these cells. But if you take BARC1 positive cells and do EGS2 knockdown, it does not actually bother to actually go into say cell death. You still see the same amount of cells over there in the colony cells. So the whole idea is, if you inhibit EGS2, the cells die. Only the cells die which are having BARC1 kind of deletions. BARC1 deleted cells are sensitive to EGS2 inhibition. So this is the kind of the prior knowledge we had, and we thought maybe I think we can combine these EGS2 inhibitors with any kind of rational drug combinations or kind of drug combinations and find other combinations which can be actually useful for kind of combinatorial therapy. And we did a drug screen. In this drug screen, we actually take many small molecule inhibitors from many classes, like cell cycle inhibitors, bunch of cell cycle inhibitors, kinase inhibitors, hyposignaling and epigenetic inhibitors. And these are based on kind of the genetic knowledge we had, because NF2 is mutated, so we choose specifically hyposignaling pathways because of the CDK into cell cycle and kinase inhibitors. And the epigenetic is a very important part, so we choose also epigenetic library. And we combine that with EGS2 inhibitors as our anchor drugs. And then we kind of vary different drugs in different concentrations, put a matrix format, and then we drug screen. So I will not go to that one. What we kind of take over from there. So we found that FGFR inhibition, the fibroblast growth factor inhibition, is a very effective combination if you combine with EGS2 inhibitors. And then I will give you some example how we actually proceed these inhibitors to the next level. But before actually finding these FGFR inhibitors, we already had a paper, so where we studied along with the Wellcome Trust and Cambridge Institute. They are actually with that institute, we have a big collaboration and do a drug screening, like combinatorial drug screening with 1,000 cell line panel. And so basically we have data of 889 cancer cells across all the tissue type. And we see that specifically in this, all the cell panels, if you look at these are the effective doses where the FGFR inhibitor is quite effective. We see a lot of mesothelioma cell lines are actually present there. And when we zoom in, we could see that these cell lines, if you look at over here, this is called the in vitro cell killing assay. Kind of say you see viability at a particular day, and you have the drugs concentration of the FGFR inhibitor. All these red lines tells that these are the BARC1 low expressing cell lines, and the blue lines are BARC1 high expressing cell lines. It means that the BARC1 low expressing cell lines are quite sensitive to FGFR inhibitors. And this we have actually published in Cancer Research. And also, if you look at over here, actually it's also worked very good in the xenograft models. So we had some prior knowledge that FGFR inhibitor is, say for example, effective to some extent. But we also knew that it was also failed in mesothelioma clinical trial as a single agent. So we thought, I think it's not going to work as a single agent. So it's our best chance to actually combine easiest inhibitors with FGFR inhibitors, and that's how actually we started some new experiments. So we looked at our easiest inhibitor treated cells, and we specifically see that in the easiest inhibitor treated cells, the FGFR pathway is highly upregulated. So if you like the fibroblast growth factor receptor signaling is highly upregulated. And also a lot of molecules which is inside this PI3 kinase AKT signaling pathway, many of these molecules belong to the FGFR signaling pathway. So this actually gives us a kind of rationale that there is something happening with this and these combinations might work. So we did kind of a lot of experiment, in vitro experiment in here. And I'm going to just show a few of these because it's going to be a lot of repeated slides. So the way we do, so we took mouse cell lines, as I mentioned, NC, BNC, NC stands for it's like that, NF2 CDK2 BNC. So this will give us a kind of a biomarker based stratification. And here are the BAP1 mutated or BAP1 wild type cells. And then we use either DNS1 or single agent, say for example, single agent easiest inhibitors, single agent FGFR inhibitors, or combination of these drugs. And if you look at these combinations, it's very effective in killing cells. It is very effective in killing cells when this kind of BAP1 mutated. It completely eradicates these cells. Maybe it's not visible that good. So if you look at this map, it actually tells this is kind of a more broader map which also has using a lot of different drug concentrations, not only a single concentration. So the mountain kind of structure, it actually tells the synergies, the synergy level in these drug combinations. But if it's kind of more flat lines, it does not work in synergistic combinations. And what we have done, we have used two different easiest inhibitors, and we have also used actually two different FGFR inhibitors, one is AZG and one is the PD1 compound. And we use specifically 5 different BAP1 proficient, 5 different BAP1 deficient human mesothelioma cell lines. And also we have taken a bunch of other cell lines which are actually having BAP1 mutations, for example, EVL melanoma or renal cell carcinoma. And we have tested this in all these combinations, and it works like a charm. To just give you kind of an animal experiment, here we have done a xenograft experiment. So in the xenograft experiment, we take either BAP1 negative or BAP1 so proficient cell lines, we transplant them into the skin fold, and then we treat, once they grow 200 millimeter cube size, we kind of treat them with these drugs and see whether the combinations or single agent does it work. And if you look at these combinations actually work like very, it kind of controls these cell proliferations as compared to the other single agent and the vehicle in BAP1 deficient cells specifically. But if you look at BAP1 proficient, it also works to some extent, but it's not really that, it's not controlling that good. So I also mentioned that I had developed a nice mouse model which you can directly test on the immunocompetent autologous models. So here also we kind of induce tumor and treat these mice either with vehicle or these drug combinations, either AZD4567 or tegometrostat. As you can see that the mice treated with these drug combinations can survive almost 92 days, but if you look at the mice which are not treated, they can die within 41 or 51 days. The median survival is 55 days. And if you look at the tumor burden also inside the thoracic cavity, drastically decrease on the treatment side. And this is kind of some kind of proof. But if you look at the treatment situation, the tumor burden is going very low, but if you look at over here, there's a lot of tumor cells in the waiting control cells. And actually we patented these combinations, and we are now planning clinical trial based on these drug combinations. And now we have tested also in many other tumor types, not only in mesothelioma. As I mentioned, we try it in the EVL melanoma cells. Again,
Dr. Jitendra?
Yeah, just
under 3 minutes.
Okay. So in this cell line also it was like a BAP1 specific manner. And in another approach, what we did, instead of drug screen, we did a genetic screen. So in that case, we used unbiased CRISPR screen to find also rational combination along with easiest inhibitors. And there we found another drugs called actually pathway called mevalonate pathway, which is actually a lipid biosynthesis pathway. If you combine easiest inhibitors with lipid biosynthesis pathway, they can also work together like each other And this is another approach you can actually also figure out rational drug combinations. So with this, I would like to tell, so these two combinations, we are now doing a clinical trial in Netherlands Cancer Institute for mesothelioma, with the two are phase 1 to clinical trials. In one case, we are using EZS2 plus zoledronic acid as a kind of a inhibitors of the nevelonate pathway, and the other one we are using EZS2 with FGFR inhibitors another arm, and conducting a clinical trial. So with this, I would like to just summarize. So as I mentioned, we have developed a mouse model which is quite relevant to study or find rational drug combinations, and through these tools we have established drug combinations such as EZS2 inhibitors or FGFR inhibitors or EZS2 inhibitors and nevelonate pathway inhibitions. And these combinations are quite synergistic, as I mentioned, these two. And we are undertaking a clinical trial based on this data. So with this, I would like to thank the organizer and also many people involved in this study. And I would like to stop and take questions.
Very good presentation. Any doubts or any questions from the floor? Yes, please. You can come to the mic.
Thank you, Dr. Jitendra, for your nice presentation. So I have just one question regarding the combination.
Yeah.
So is it a synergistic effect or additive effect?
It's a synergy.
But for a different mutation, you also showing that. So that kind of thing, there is a graph pattern is different. So based on that, there is this depends on the mutation kind of thing.
So as I mentioned, this is a biomarker based type. If you have a mutations in BAP1 or if you have defective protein in BAP1, only it works in the BAP1 deleted situations. So this drug combination is only effective in a BAP1 deleted situations. It could be any tissue type. It could be mesothelioma, it could be uveal melanoma, it could be renal cell carcinoma. So the BAP1 is our biomarker as a kind of to stratify patients, and then we can actually go for this clinical trial.
Thank you.
Any more questions?
Thank you, Doctor. This was a really nice presentation and very informative as well. So one quick question regarding your combination therapy.
Yeah.
The question is like you did this combination therapy in cell line model or it was a mice model? The second thing is like when you used a different class of inhibitor, whether it is TKI or any other class.
Yeah.
So what was your combination matrix? Like it was a co-treatment with your BAP inhibitor or it was a sequential treatment? If you can throw some light on that.
Yeah. So actually, what we have done in case of say epigenetic, most of the epigenetic inhibitors, they takes time before actually it works. Epigenetic changes does not happen like just within 1 or 2 days. So what we do in case of in vitro experiment, we kind of pre-treat the cells with EZS2 inhibitors for a certain amount of time, which is actually roughly 5 to 7 days. That was the kind of time, and then we use the second drugs. This is for in vitro strategy. But for our in vivo strategy, we haven't actually used that strategy, but we still kind of use both the drugs at the same time, and still that is effective. So in clinical trials, what we are doing, we are not doing sequential, we are still doing it kind of a combinations together.
Any more questions from the audience? Thank you, Dr. Jitendra.
And thank you so much. I just want to have a small announcement. So as you mentioned that as you can see, we are kind of doing a lot of say clinical related research and health data science research, and together with one of my founders, we have founded a company called SciMedian, and we are training young people from India to actually get a good grasp on next generation sequencing and health data analytics. If anybody interested, you can go to our booth and have a kind of talk and see what we can offer. We would like to mentor people and get them actually to the job market in health data science field.